Hey students, this is Mrs. Foy, and this is a brief introduction to cancer. So by now you should have a good understanding about mitosis and understand that the cell cycle and what's going on with mitosis has a lot to do with cancer. So there are stop and go signs that have an impact on how the cell goes through the cell cycle. And those are both internal and external signals that the cell gets. So an example of an internal signal would be when those kinetochores do not attach to the spindle microtubules. And what is supposed to happen, as you remember, the kinetochores are kind of a protein bracket that sits around the centromere of the chromosome, and that is where the microtubules attach. Now, there are some non-connecting kinetochores that lengthen during mitosis. There are actually lengthening those spindle fibers, and they are not attached to the kinetochores. But there are other spindle fibers that attach to the kinetochores, and those are shortening. They actually shorten those protein strands of the microtubules to pull the chromosomes apart. So you have some that are not attached to the kinetochores that are linking, and some that are attached to the kinetochores that are shortening. And so those push and pull the chromosomes to the opposite poles, and that's what happens during anaphase. And there is supposed to be a checkpoint of an internal signal that when the spindle fiber that's supposed to be connected to the kinetochore is not connected, then there is a signal that delays anaphase so that the cell will not divide and produce daughter cells that don't have the correct number of chromosomes. An example of external signals that are going to affect the cell cycle would be growth factors. And growth factors are proteins that are released by certain cells. Sometimes they are hormones that stimulate other cells to divide. And you can think of those as ligands that come in and attach to a receptor, and then a signal transduction pathway ensues after that that ends up turning the cell on and having the cell divide. For example, one type of growth factor is called a platelet-derived growth factor, or PDGF, that stimulates the division of human fibroblast cells and culture. So fibroblasts are cells that are that produce certain types of connective tissue in the body, and we can grow these. We have learned how to grow these in culture. So we know that if you have some cells that you have on a petri dish and you put it in some medium that has some nutrients and special things inside the nutrient to make it grow, that if you don't have this particular PDGF growth factor, the cells do not grow. But if you do have it, and it's actually this color, it's kind of a pretty kind of gold color, then your cells do divide. So we know that that works in vitro. So here's a good vocabulary word for you. In vivo is the Latin word that means actually in the cell, and in vitro would mean something that occurs outside the body. So another example of an external signal is density-dependent inhibition. And we've already talked about the fact that normal cells, when they get so crowded that they actually touch another cell, that would set off a signal that would then tell the cell to stop dividing. There's no more room for more cells. Another type of cell external signal that most animal cells have is something called anchorage dependence. So this is the idea that the cell cannot usually grow if it's just floating around. It actually has to have some kind of a some type of a scaffolding system that the cell can actually adhere to and grow on. So when scientists are trying to grow tissue cultures, they actually have to put a substratum or some type of scaffold for the cells to grow. And that's very important for cells that are growing. Most animal cells need that. But cancer cells don't have anchorage dependency. And cancer cells don't have density-dependent rules that they follow. And they don't respond normally to the body's regular control mechanisms. And as a matter of fact, some cancer cells don't even need growth factors to grow and divide. And this may be because, number one, they make their own growth factor, or number two, they convey a growth factor signal. So they kind of fake out the cell by making something act like a growth factor that's not really a growth factor, or they have an abnormal cell cycle system controlling. A normal cell that's converted to a cancerous cell is a process called transformation. And this is a little confusing, this word transformation, because we actually use it in two other biological terms. So when you talk about transformation, you need to understand the source. This is a normal cell becoming a cancer cell. So cancer cells are going to form tumors and masses of abnormal cells that are within other normal tissue. And the problem they cause is a little bit like weeds. So you have a situation where you have, if you were trying to grow a plant and you had a bunch of weeds around that plant, the problem is that the weeds are going to get the sunlight, they're going to get the nutrients, they're going to get the water, and they basically starve the plant that you're trying to grow. And that's very similar to what's happening with cancer. Cancer will take nutrients, it will take resource, it will take space, and basically starve out the normal cells, not to mention causing pain. If abnormal cells remain at their original site, then the lump is called a benign tumor. But a malignant tumor is a tumor that has the ability to kind of send these offshoots of cells into other parts of the body, which we call metastasis. So it's exporting cancer cells to other parts of the body where they will then form secondary tumors where they land. So here's an example of breast cancer metastasis. And we can see that as the cancer in the breast tissue starts to become more advanced, at a certain point, the cancer cells can metastasize either through the bloodstream or through the lymph system into other parts of the body, and they can then form secondary tumors. And interestingly, different types of cancer usually have very specific secondary metastasis sites. For example, prostate cancer often metastasizes to the bone, and other types of cancer metastasize in other places, which are characteristic of that particular type of cancer. So we know that obviously early detection is one of the main life-saving treatments that we have for cancer, and that is a system of categorizing how advanced the cancer is. So cancer that is a stage 4 cancer has already metastasized to another organ in the body. And again, this can be through the lymphatic system or through the blood system. And this is a picture of colon cancer that has grown from the polyp stage. So you may have heard of people that have polyps in their colon, where it is actually invading deeper and deeper types of tissue, lining the colon until it becomes stage 4, where it would actually invade another organ. So one of the most interesting and tragic cases of modern science actually has to do with a woman named Henrietta Lacks. And Henrietta Lacks was a poor African-American woman in the 1950s that lived in the South, and she actually developed cervical cancer, went to her doctor, and the doctor took a sample of her cervical cells for a biopsy, which they normally do. And at that time, this was actually at Johns Hopkins Hospital down in Baltimore, they attempted to culture these cells in an attempt to learn more about cancer. And at that time in the 1950s, they didn't know how to grow cells very well in culture. And so these particular cells from Henrietta Lacks grew. And so this was great because they were growing in petri dishes and culture dishes. And so the pathologist and the people that were working there at Johns Hopkins began to send these cells to other labs in other parts of the country and the world who were studying cancer, who were trying to learn about cancer and about cells and culture. And the code for these cells was called HeLa cells for Henrietta Lacks. And these cells became basically the workhorse of cells in the biotechnology labs around the world. HeLa cells are available today. You can actually, I have ordered HeLa cells from biological supplies companies. And these cells are immortal. They continue to divide and divide and divide. And these were cells that were from the cervical tissue of Mrs. Henrietta Lacks, who died very shortly after these samples were taken. And so her story is a very interesting one about ethics and research and the idea of who owns tissues and who owns biological properties and things. And so there's an excellent book about her life, and you should see me if you would like to know more about this, because it's a really, really great story. So these are pictures of HeLa cells under the microscope that were originally the cells from Henrietta Lacks' cervical cancer. What are 3 genetic malfunctions that lead to cancer? So usually what happens is you have multiple genetic malformations or mutations that would lead to cancer. Some of these are mutations in accelerator genes, mutations in brake genes, and particularly mutations in what we call the gene P53, which is the guardian of the genome. This is kind of the spell checker for the body, and interestingly enough, this mutation is found in about half of human cancers, this P53 mutation. So first we're going to talk about mutations in accelerator genes. So accelerator genes message is to grow and divide, and these are called proto-oncogenes, and their job is to they are going to code for proteins that develop receptors that respond to growth signals, and then through signal transduction, that's going to signal the nucleus to divide. Mutated accelerator genes are called oncogenes, okay? So normal accelerator genes are proto-oncogenes, but mutated accelerator genes are called oncogenes, and these can lead to cancer. 2 examples of oncogenes are SRC and RAS. Mutations of brake genes can also lead to cancer. So in a normal tumor suppressor gene, these are going to lead to signals that slow down cell division. So a mutation in a tumor suppressor gene can also lead to cancer. And this guardian gene, this P53 gene, is a very important gene, because one of the things that it does is it's going to survey damage in a DNA or damage in a cell, and then it will program that cell for apoptosis, or program cell death. And so mutations in this P53 gene disable this programmed apoptosis, this programmed cell death, and allow cancer to start. So this is a diagram that shows just some of the signals that are coming in through cancer. For example, here's a receptor that would be coded for by an accelerator gene, okay, or a proto-oncogene, and you can see that the growth signal coming in, or these growth factors, would then bind to this receptor, and then you would have a signal transduction pathway that would then signal the nucleus to divide. Also, we have receptors that are from tumor suppressor genes that would respond to signals that say, stop dividing. So one of the things that cancer researchers are looking for are drugs that would then block these receptors, which is very important. Future cancer drugs may also activate the cell death program and stop cancers from continuing to divide. Another cause of cancer that we know of from our research has to do with the ends of chromosomes called telomeres. So we've talked about centromeres, which is the waist of the chromosome, but telomeres are the ends. And one of the things that we know that we've learned about telomeres is that there are repeating sequences of DNA that do not code for proteins that are on the ends of chromosomes, kind of like those plastic things on shoelaces that keep them from fraying. And every time a normal chromosome divides, the telomeres lose a little bit of the DNA that's on those ends, and they shorten. And at a certain point, the research shows that when these telomeres get to a certain point where they're so short, then they do not divide anymore. It's kind of like that's the life of the cell. But cancer cells are able to turn on genes that produce telomerase. Telomerase is an enzyme that actually rebuilds telomeres. And so in an insidious way, they are actually becoming immortal by continuing to fix their own frayed shoelaces, so to speak, and so makes their telomeres not shorten. Angiogenesis is the production of new blood vessels. So angio means blood vessels, and genesis, of course, means new. And one of the things that happens with cancer cells is that they have to get a blood supply because they need nutrients, they need oxygens, they need to get oxygen, they need to get rid of their waste and their CO2. So here you see these pink cells are normal cells that have a blood supply. But these purple cells are cancerous cells, and you can see that there are a bunch of new blood vessels that are supporting that cancerous cells. So one of the things that happens with cancer cells is that they can produce a protein called VEGF, and VEGF is going to send out a growth signal to cells that line the organs. These are called endothelial cells. And what happens is those endothelial cells then, because of this VEGF growth factor, are going to form blood vessels, and these blood vessels then are producing a blood supply to the cancer cells. And so scientists are testing different anti-angiogenic proteins that are either going to block the relay of these VEGF growth factors or stop the growth of endothelial cells to keep the cancer cells from making their own supply chain. We also know, of course, that heredity plays a huge part in cancer's environment that we know causes cancer. We know, for example, but we can also inherit mutations from our parents that give cancer a jump start. So that was a picture actually of cancer. And this is a picture of what can happen in kind of a domino effect that somebody might inherit, for an example, a mutation in one of the genes that might not by itself cause colon cancer, but the cumulative effect of inheriting these genes from parents and also from the environmental damage that occurs can lead to several mutations that would lead to the development of colon cancer. And we know, for example, that in this particular type of cancer, it may take 50 or more years for these different mutations to accumulate and the start of cancer to begin. There is an amazing connection between genes and cancer. And this is a website, for example, run by the NIH that is looking at genetic markers for very specific types of cancer. This is a very exciting area of research because basically what we know is that breast cancer is not breast cancer. There are lots of different forms of breast cancer. And if we can do genetic testing on the cancer and find out specifically what type of markers are specific to that cancer, then the chemotherapy can be specifically tailor-made to that particular type of cancer based on the genes, the genetic fingerprint that that cancer is showing. So that's really, really exciting news. Also, scientists have virus, the same virus, because we know it's some and that's exactly cancer and to prevent cancer and